you're welcome to this lesson. It is lesson number 4 of soil. Now, this one we are going to talk about soil living organisms. In the previous lesson, you have talked about those other components of soil: water, humus, and mineral salts. Now, in this particular lesson, we are going to talk about soil living organisms. Now, soil living organisms are divided into two. We have microorganisms and macroorganisms. Microorganisms, these ones are microscopic. They cannot be seen by our naked eyes. And they include bacteria and fungi. Remember, these microorganisms, they play an important role in maintaining soil fertility through decomposition of plants and animal remains. When you die, or when any other animal dies, those bodies are supposed to be decomposed by the bacteria to form humus. And I think you saw the importance of humus to plants and even to the soil. Now, the second group of organisms, we have the macroorganisms, and these include the earthworms, the soil arthropods, and many others. Now, earthworms are common in moist soils, which are rich in humus. By the way, these earthworms, they dislike dry or acidic soils. Now, let's move to the next slide. This slide is showing us some of the earthworms. You can carefully look at these diagrams. They are showing the different, they are showing earthworms. And I think these earthworms are in the humus. Now let's go to importance of living organisms. And now this importance, therefore, to the soil actually. Now, living organisms, we realize that they improve on soil fertility, like we have seen in the previous slide. The bacteria will decompose the dead organisms to form humus, and also the nitrogen fixing bacteria will convert the nitrogen in the air into the nitrates, and all this improves on the soil fertility. Soil living organisms like the earthworms, they burrow in the soil, and this improves on aeration and drainage. They make some tunnels through the soil. By the way, whenever it rains, we usually see these earthworms coming out, and by doing that, these earthworms will be creating some tunnels in the soil, and that one will enable the soil to be easily aerated. The next one, some of these living organisms in the soil can cause diseases to man and his plants. Do you realize that there are some diseases which are caused by bacteria? And these diseases can affect both the plants and at the same time man. Now, the wastes from soil living organisms add fertility to the soil. Now, which wastes are these? These wastes can be from these earthworms and also from other organisms. When they decay, they form humus and eventually improve on the soil fertility. Now, on our next slide, we have some video. I think you can watch this video. You try to tap on that link. You'll watch a video about those living organisms. Now let's move to experiment to investigate the presence of living organisms in the soil. Now here, when you look at the materials we are supposed to have two conical flasks. We are supposed to have muslin bags, 2 of them. Then we are supposed to have a soil sample. And this soil sample, we get the topsoil. Then we are supposed to have 2 corks and lime water, or the bicarbonate indicator. Now, lime water and bicarbonate indicator, they play the same role in this particular experiment. Let's move to procedure. In the procedure, we are supposed to collect a handful of fresh topsoil and divide it into 2 equal portions. For example, we can get soil we can get topsoil and then we divide it into two equal portions. We can just measure this either using a measuring cylinder or a balance. Now after that, we are going to label these portions A and B. Now what we shall do, we are going to sterilize one portion of the sample, and in this case it is going to be sample B, the one we labeled B. And how are we going to sterilize? By heating it strongly on a crucible for about 30 minutes. Now after heating for that time, we shall leave this sample to cool and place it in a muslin bag. Muslin bag, I can take an example of these white buveras. I think I can take that example. I think you're more familiar with that one. But when you come back at school, we shall show we have bags. Now, the next procedure, we shall place the remaining portion of the fresh soil sample in another muslin bag. Remember, I told you that we are supposed to have two of them. We are supposed to have two muslin bags. So we have used one in the second procedure, and then we are using the second one in the third procedure. Now, after that, we are going to add equal amounts of lime water or bicarbonate indicator solution in the conical flasks, and then suspend the muslin bags with soil in the conical flasks as shown in the setup below. We are going to see this in our next slide. Now, after this, we are supposed to allow the setup to stand for about 2 days, and then observe the appearance of the lime water or the bicarbonate indicator solution. Now, let us take a look at our setup. In this setup, we have setup A, and we have setup B. Now, in setup A, we have a muslin bag inside the conical flask. We have a muslin bag, and inside that muslin bag, there is fresh soil sample. And then in that very conical flask, we are having lime water. Now, when you look at setup B, in the muslin bag, instead of having fresh soil sample, we are having heated soil sample. And then also having lime water or the bicarbonate indicator. So you can look at this setup critically before you continue the next slide. Okay, now let's move to the next slide. On our next slide, we are having observations. What do we observe after that period of time? What do we see? Because you make observations after the time has elapsed. Remember, our time was 2 days. Now, what we are going to observe, we are going to realize that the lime water or the bicarbonate indicator in conical flask A turned milky. That is for the case of the lime water. But for the case of the bicarbonate indicator, this bicarbonate indicator solution is going to turn yellow. So what does that one mean? That one means that in this particular setup A, I think there was some gas that was liberated, and this gas is expected to be carbon dioxide gas. We know carbon dioxide gas is the one which turns lime water to be milky, or it can turn the bicarbonate indicator to become yellow. Now, in our conclusion, we realize that carbon dioxide was produced in setup A during respiration, indicating the presence of living organisms, whereas in setup B, the lime water remained clear. Why? Because the living organisms were killed by heating. Remember, we sterilized the soil sample which was placed in setup B. That's why in our conclusion here, we are not seeing lime water turning milky. Now, let's move to the next slide. But before we move to the next slide, don't forget the explanation for that particular experiment. Why the other one is turning milky and this one is not turning milky, or it is remaining clear. Now, let's look at types of soil. Soil is grouped based on size and nature of the soil particles. On this basis, there are only 3 types of soil. The first type is clay soil, the second type is loam soil, And then the last one is sandy soil. Now let us first begin by talking about sandy soils. Now sandy soils contain large spaces between the particles, and these spaces allow water to drain off very quickly. By the way, let me try to tell you more about this sandy soil. How can you get this sandy soil? Where can you get it from? Sandy soil, we can easily obtain it from the roads where we pass. These parts where the water passes, those parts, that's where we have sandy soil. Now, let's look at more facts about sandy soil. They feel gritty. They have a gritty feel when wet and felt between the thumb and the finger. That word there is finger, not figure. Now, when you wet sandy soil and then you feel them between your thumb and the finger, they are going to have a gritty feel. They contain only very small quantities of water, and they may be deficient in calcium and magnesium. By the way, these soils are described as light soils because they are relatively very easy to work with. Let's move to the next slide. Now, on our next slide, you will see some sandy soil. Now, when you look at this picture here, you realize that when you go to construction sites, I think this is what they use for constructing these houses. So you try to look at that sandy soil. Please don't forget to touch it. Don't forget to feel it between your finger and the thumb. Now, our second type of soil is clay soil. Now, these ones, they have small fine particles. In agriculture, you talked about texture, soil texture. Now, clay soil has a fine soil, it has a fine texture. Now, the clay soil particles are closely packed. These ones are closely packed together just because it has very small particles. They are closely packed together, and this one leaves very small spaces between them. By the way, when they are closely packed together, this one is going to cause clay soil to have poor water holding, to have poor water drainage, and eventually this one can make the soil to be waterlogged. When soils that are waterlogged, they are these soils which cannot easily lose water after it has rained. The clay soils are very difficult to work with, and therefore they are described as heavy soils. Then they have a sticky feel. A sticky feel when you feel them between your thumb and please don't forget, whenever you're feeling these soils, don't forget to wet them. Now, when lime is added to clay soil, the clay soil particles aggregate. The clay soil particles aggregate together to form larger particles. This helps to improve aeration and drainage. Remember, the challenge of this clay soil is a lack of what? The clay soil particles are packed, they are closely packed together. Now, when you add in lime, they aggregate together. And this, like we have said, it helps to improve on aeration and drainage, and it also reduces on acidity of the soil. And that process is called flocculation. Let's move to the next slide. Now, on this next slide, we realize that the clay soil is trying to, what is happening to clay soil after it has rained. You see it is having some. Now you see on the next slide, after adding lime, that is what is happening. Now let's move to loam soil. Now loam soil is a mixture of sand, silt, clay, and organic matter. It has a stable crumb structure and is the best for crop production. Even when your home, in your gardens, even when you have clay soil, you have sandy soil, you can improve that soil and you carry out agriculture. How? By adding in humus. I think in agriculture you've discussed much about humus, how you can easily form humus, and in the previous lesson of Mr. Jonai, you discussed the importance of humus. Now when you add humus to sandy soil, to clay soil, you will easily carry out your agriculture, and the produce will be much than you expect. So here we are having some humus. Now let's go to the next slide. On our next slide, we are looking at the differences between clay soil and sandy soil. I told you where you can easily obtain this sandy soil. For the case of clay, you can go to the ant hills, you can get that soil there. So you compare that one from the road and this one from the ant hill. We shall realize that this one from the ant hill has very small air spaces between the particles, whereas the other from the roads, it has large air spaces between the particles. The second one, the clay soil, is rich in dissolved salts, whereas the other one is poorly. It is having low dissolved salts. Then we have high water retention capacity. Then the other side it has poor water holding capacity. Then plus these other differences, like you can see from that slide. Now let's move to the physical properties of soil. Physical properties of soil. And here we have a YouTube video which you can watch about the soil properties. Now let's move to the next slide, where we have the first physical property, and that is porosity. Porosity, this one is talking about the spaces in the soil. Now you will realize that sandy soil possesses large spaces between the soil particles, and so these sandy soils are considered to be more porous. It has a lot of spaces. When it comes to clay soil, these ones they possess very small spaces between the soil particles, and thus for it is less porous, whereas for the case of loam soil, for it is moderately porous. That is about porosity. Let's move to air content now. Sandy soil contains a lot of air. Why? Because it is well aerated. Remember, we have said it has large spaces, and those spaces can make the air to pass through those particles easily. Then for the case of clay, for it contains little air, so it is considered to be poorly aerated. And I think when you look at porosity, you can easily get the reason as to why it is poorly aerated. Then for the case of loam soil, this one for it is just in between. It is in between clay and clay. It is in between clay and sandy soil. So for it is moderately aerated. Now let's talk about drainage of water. Now when it comes to drainage of water, don't forget the spaces we have been talking about. Sandy soil has good water drainage, so it allows water to pass through it very quickly. Now why do you think it is like this? It is just because of those spaces. Then clay soil has poor drainage of water, and this makes clay soil to be waterlogged. I've already talked about this, and it can be improved by adding in humus. By the way, when you were discussing about the importance of humus, we realized that this humus can stick these soil particles, can aggregate the soil particles together. And of course, once these clay soil particles are aggregated together, then we can improve on the drainage of water in that particular soil. Then loam soil, for it drains water moderately. Now let's go to water retention capacity. This one is the same as the water holding capacity. It refers to the amount of water soil can hold. Now we realize that sandy soil holds little water, so it has poor retention capacity. Still, the reason is about the air spaces. Because of the very many air spaces in sandy soils, so water can easily drain through. So it has poor retention capacity. And it can also be improved by adding in humus. The humus will bring in the particles together, and eventually the drainage, so the water holding capacity will be improved. Then clay soil tends to become waterlogged, and it holds a lot of water, so it has high water retention capacity. The reason we have already talked about that. Then loam soil holds water moderately, but not becoming water logged, does not easily become water logged, so it drains water. Actually, it is the best for agriculture because of that reason. Now let's look at experiment to compare drainage and retention or water holding capacity of water in sandy and clay soils. Now when we look at this particular experiment, we have two in one. We are looking at drainage, looking at the water holding capacity. Now here we have the materials. What are we supposed to have in this particular experiment? So the first material, they are the filter funnels. Now I have an example of a filter funnel here. When you look at this filter funnel, I think you can look at it clearly. We are supposed to have two of them. We are supposed to have two of them in this particular experiment. And if we are comparing water holding capacity or drainage in 3 soil samples, then we are supposed to have 3 of these filter funnels. Then we are supposed to have measuring cylinders, but in this particular experiment we are supposed to have 2 of the measuring cylinders. We are supposed to have filter papers. I believe you have seen these filter papers. Now the next material, they are the soil samples. We are supposed to have dry sand soil and then dry clay soil. Remember, I told you where you can easily obtain these soil samples from. Please, at home you can even perform this experiment. Please be scientific. Biology, we can do biology anywhere. You know, biology is living, and wherever you go, you see life. Now the next material is water. I can show you water. Water, I think it is even in your homes. You can easily obtain the water. Then the beakers. So this is an example of the beaker. So in this particular experiment, we can have 2. If the experiment is about 3 soil samples, we are supposed to have 3 of these ones. So those are some of the materials. Now let's go to the procedure. Procedure number 1, we are supposed to measure equal volume of each soil sample. Remember, we have 2 soil samples, the sandy soil and clay. We are going to use the measuring cylinder for measuring. We shall measure using the measuring cylinder, and in this case, I can take an example whereby measuring 50 cubic centimeters. 50 cubic centimeters of the soil sample. I measure for sand, and I also measure for clay. Remember, I told you that we are supposed to have 2 of these ones. Now, after that, we are supposed to fold the filter papers properly and put 1 in each funnel. Remember, I told you that this one is the filter funnel. After folding our filter paper, we insert that filter paper here in the filter funnel. Now, after that, we are supposed to place clay soil sample in this filter funnel, and also the sand in the second filter funnel. And after that, we are going to place the filter funnels plus their contents on the measuring cylinder. Now, after that, we are going to measure equal volumes of water, and then we are going to pour that water in the soil sample, of course, from this side of the filter funnel. From this side of the filter funnel. Now, after pouring, of course, you pour in this one, and you also pour in the other one, which is containing sand. After that, we shall allow the setup to stand for some time. For example, like around 30 minutes or 20 minutes. Now, this is the setup of our apparatus. So we are having sand, and then we are having clay. Now, after that, we are supposed to observe and see what will happen. Now, when you look at this, we are seeing that water is going through into the measuring cylinder. The water is going through the sandy soil and clay soil into the measuring cylinder. Now, here we are comparing these 3 soil samples: sand, loam, and clay. Now, in our next slide, we have observation. From this observation, we realize that water passes through sand soil faster than clay soil. So much water is collected in that cylinder, which is having sandy soil, and less water is collected in the cylinder containing clay soil. So in our conclusion, we realize that clay soil holds more water than sandy soil, and therefore we can conclude and say clay soil has good water holding capacity, but at the same time it is having poor water drainage. And for the case of sand, it is having a poor water holding capacity, and it is the one with the best drainage. And all the reasons as to why it is like that, we have already talked about that. Then there is a video there which you can try to watch. Then when we move to the next slide, this one is talking about the capillarity. Capillarity through soil means how well water can rise up in the soil, and this depends on the size of the spaces, like we have discussed about this, between the particles. Sandy soil has the lowest capillarity because of the spaces, I think, and then clay has the highest capacity. So in the next lesson, we are going to talk about